So, welcome to this class on receptors part 2. In this class we will be talking about Golgi tendon organs, what are called as joint articular receptors, and some cutaneous receptors. In the previous class we saw the case of muscle spindles and how muscle spindles serve as length and velocity sensors. So, in today's class we will see a different sensor. So, if a note, in the case of muscle spindle we said that the sensor itself or the intrafusal fiber that is responsible for sensing the length is aligned in parallel with the force producing extrafusal fibers. This was the mechanism. But is there some other way in which I can know, is there some way in which I can know the force produced by a muscle? Can I know the tension in the muscle? That is the question. The answer is yes, and the sensor that is responsible for the detection of tension is called as Golgi tendon organ. So, how does this do that function? Golgi tendon organ is composed of a capsule that is located between the muscle and the tendon, or located near the junction of the muscle and the tendon. To remind ourselves what this situation is, so let us say I am going to draw two bones. So, that is a bone representationally. So, there is another bone here, and we said that movements are produced by relative movement of bones, right? Not. Let us say that is the muscle, and so this is called as a muscle or the muscle belly. This is composed of contractile material and is neurally innervated, and the muscle is attached to the bone via tendons. This attachment, so what are the tendons composed of? They are also composed of contractile material, mainly they are composed of collagen. This muscle, when it contracts or when its length reduces, that means that will happen when the neural activation makes that to contract like that, for example. Then what will happen, either the bones will move relative to each other or the tendon will elongate. Okay, this is the process. In a different class we will discuss the details of this. So, the tendon is attaching to the muscle at that point. So, what you are seeing here is a zoomed out version of that. So, if we zoom out that, on the one hand you have the muscle fibers, on the other side you have the tendon. Okay. So, I am zooming out that part where the muscle is attaching to the tendon. In between the attachment you have a capsule that is composed of fibers that is intertwined by a neuron, axons of a neuron. This neuron is, so this axon is called as a 1B afferent axon. We saw that the 1A afferent axon is the primary spindle ending, and 2 is the secondary spindle ending, is it not? When this is getting pulled, suppose this tendon is attached to a bone here, this is getting pulled, when that is getting pulled, gets compressed because of the collagen fibers. So, the collagen fibers become uncrimped, the collagen fibers elongate and compress the neighboring 1B axon. As the tension in the muscle increases, the compression of the collagen fibers increases. Because the compression of the collagen fiber increases, the number of channels going to be affected by the compression of these collagen fibers in this 1B afferent axon is going to increase, producing action potentials. So, what does this mean? When will this fire? That means whenever there is an increase in tension of this muscle, there will be an increase in the firing rate of this 1B afferent axon of this neuron. So, because of this reason, Golgi tendon organs are nearly perfect force transducers, nearly very good force sensors. And a question is, how do we know this? It turns out that there have been experiments on animals where they have studied the force versus say firing rate or the response rate or whatever. And for different in different Golgi tendon organs. So, as the force increases, so there are, that is not there. So, with different slopes, but in general what you will see is that, you know, as the force increases, the firing rate increases. So, if you take an ensemble of all these things, you are going to get something like that. So, this is a relatively accurate force transducer and with some slope, right. So, if we know what the slope is, then I am going to be able to estimate the force depending on the firing rate if I know the approximate slope. So, I am going to be able to do that, but it is not just one Golgi tendon organ, it is multiple, is it not? There are many. So, information is coming from many such Golgi tendon organs. So, also there have been studies. So, another way of representing the same data is that, you know, if the muscle force is at some baseline level, there is going to be some baseline firing rate for that. As the muscle force keeps increasing like that, you see that there is an increase of firing rate. As the muscle force is kept constant at a different level, there is continued increased firing level here, and then as the muscle force is reduced, there is a reduction in the firing rate. So, then again once again telling us that, you know, this serves as a very good, a relatively accurate force transducer. Another question is that going to be consciously sensed? Now, that means, can I say exactly how many newtons is that? It turns out that the debate on the conscious proprioception versus a non-conscious proprioception is not yet settled. At least in the case of the Golgi tendon organs, this information is not consciously felt by the organism. How is it even being used? What is the use of this? There must be some use, but when we say conscious perception, we are not able to say I am producing so much force, whereas in the length case, it is useful to, with the help of other receptors also, it is useful to come up with a body sense, like where my body parts are. Here in this case, how much force I am producing, the people are not able to consciously say that. So, it seems, or at least the current view is that at least in the case of Golgi tendon organs, this information goes to processing of movements, future movements, planning, processing and correction of the present force produced on the environment, etc., but not necessarily on to the conscious level. Where does all this information go? So far we have seen 3 cases. We have seen the 1A or the primary spindle ending, we have seen 2 or the secondary spindle ending, and then we have seen 1B, which is the Golgi tendon organ, is it not? Where does all this information go? These 3 together constitute what are called as proprioceptors, or some sense of body position or body's relative position, body parts' relative position with respect to each other, right. So, where does this information go? Depending on where this information is coming from, they can go to several places. These pathways we will discuss in future, but at least I will just introduce in this class. Information from these, we said that these are sensory axons, these are sensory neurons, is it not? We said that this information will go to the dorsal column. The neurons on the ventral side are usually the motor neurons, the neurons on the dorsal side are usually the sensory neurons. So, this information goes to the dorsal spinal cord, there in the dorsal spinal cord, that is like saying this is going to some place in the body. So, in the dorsal spinal cord there is a nucleus called the dorsal nucleus of Clark. Depending on, again this is depending on where from this information is coming. If this is coming from thoracic vertebra level 1 to lumbar vertebra 4 or 5, let us remind ourselves the vertebral column and its classification. There are 7 vertebrae in the neck, these are called as a cervical vertebra, and there are 12 vertebrae in the thorax and about 5 in the lumbar region, and then depending on classification sacrum is either 1 vertebra or 5 vertebra. So, if the information is coming from T1 to L4 L5, then it is going to the dorsal nucleus of Clark, and from there is a path that takes it to 2 places, one to the cerebellum. So, already we are talking about brain structures. I will be referring back to this in future. We will explain this function in greater detail in future classes. Information goes to 2 places, one is the cerebellum, the other is the primary somatosensory cortex. Taking a position on what is what, in general it is believed that when the information goes to the cortex, it is more consciously perceived, and if it is going to subcortical structures, it is not consciously perceived. This is the general view, although this view is being challenged. So, information goes to the cerebellum via the spinocerebellar pathway. Okay. So, it also goes to the somatosensory cortex, where conscious perception of what is going on is going to be formed. So, so that means this information is branching into 2 places. One place where the action depending on the input is going to be taken without regard to whether that is consciously approved or consciously acceptable. Another place where that is going to be consciously processed and a decision may be taken about whether that is and if it is coming from above T1, what happens? So, if the information comes from C1 to C7, 2 specialized nuclei called cuneate nucleus and gracile nucleus. So, and then again this information goes back from these 2 nuclei, this information goes to the cerebellum and the primary somatosensory cortex. So, this information again can be perceived. What is the pathway through which this happens? Cuneo-cerebellar pathway, for example, in the case of cerebellum. So, these details and these pathways we will reserve for future classes. The point is that this information is going to higher structures through specialized pathways, and there are specific nuclei that are responsible for relaying this information properly to specific places in the brain. Then there are a group of proprioceptors that are believed to give information about joint angles. Why have all these length receptors? These 2 length receptors in some sense is going to help me detect the joint angle, is it not? So, if this is the elbow joint, say this is the forearm, this is the upper arm of a person, and suppose that is the elbow joint, and if I know the length of this muscle, then in some sense I know what the joint angle is it not? If the length is say smaller, then the joint angle is different, or if I know the lengths of all the muscles that innervate this that are attached to this joint, then I can somehow estimate, roughly at least in some sense, estimate the joint angle. The goal is to understand what the relative positions of the body is it not? And in the case of the elbow joint, I can do that, I can extend to this point, but beyond this point if I extend, what happens? It could cause an injury, right? So, at these intermediate joint angles, this sensor is going to be practically silent, but when I am at the extreme, this joint receptor, this sensor is going to start making action potentials to warn the system somehow of impending danger. You are at your extreme, do not push yourself further in some sense. So, that is the goal. So, the physiological extreme, only at the physiological extremes they are more active. We are not able to come up with a joint angle versus action potential curve in this case. So, it seems like these are in some sense measure of joint angle, but how exactly they do that is still not understood. And then we have 4 receptors especially dedicated to hand and finger function, right. These are Meissner corpuscles, Pacinian corpuscles, Ruffini endings and Merkel disc, and then there are other free nerve endings and thermoreceptors etc. not mentioned. So, when you have so in the skin you have the outermost covering, that is the epidermis, that by itself is composed of 3 layers, and below the epidermis is the dermis, and below the dermis is the endodermis, and below the dermis you have these Pacinian corpuscles, those that are in green here, and then there are these Meissner corpuscles that are here, and there are Ruffini endings here at the dermis, at the region at that level, and then there are these Merkel discs. Why have so many? Because kind of things that we do with the hand and the fingers. What are the kind of things? Several things, right. We could move our fingers on a surface like that and try and understand texture. That is one thing. I can say this is a smooth surface versus taking this keyboard and and doing that relatively rough surface, right. At least there are indentations, at least there are structural ups and downs on that, right. Another thing that we do is press hard on a relatively sharp object. When would we do that? From time to time. For example, in the case of blind people reading with braille, for example. So, there are specific indentations, specific races in placed in a particular spatial distribution, helping them to read things. So, that is that. So, Merkel discs are specialized for vertical pressure on skin surface. So, that means they are going to be very useful in the reading of say braille. Why did I say this? Why did I mention braille? Because it turns out that in the case of congenitally blind people who cannot see from birth for whatever reason, these individuals have increased sensitivity for these touch receptors, and that level of sensitivity a person with vision can never reach, right. So, because our we depend more on vision for information, whereas the blind people, because they are blind, they cannot depend on vision. So, their brain is adapted to sensing with these sensors more, and it has been shown that there is quite a bit of data showing how their plasticity at the level of brain, using neuroimaging, using different modalities, at the level of structures in the brain, there are differences between age match controls and congenitally blind individuals and this. So, while performing the same function, while doing the same thing. So, and Meissner corpuscles, in this case sensitive to quickly changing pressure. Ruffini endings, again these are slowly adapting. They are responsible for sensing larger skin areas, sensing over larger skin areas, and Pacinian corpuscles are rapidly changing. So, are responsible for rapidly changing stimuli like vibration. So, 2 of these are slowly adapting, 2 of these are rapidly adapting. Their functions are different, their differences in structure is due to the differences required in the function. Okay, so, this is the, and then there are free nerve endings, and there are nociceptors, those that sense pain, right. Those that sense pain are called as nociceptors, and those that sense temperature, thermoreceptors. There are others that we are not discussing. So much for the cutaneous receptors. Information from the cutaneous receptors is also used in controlling movements, especially of the fingers, because I have the fingers and how much the skin has deformed, depending, that is one information, at least one source of information for controlling finger movements, but that is not the only source. Obviously, there are proprioceptors of the muscles that control the finger movements, but the fingers and hands also are bestowed with these high quality receptors, sensors that are responsible for sensing a whole number of things. Note that there are only two parts in the body, two specific areas within the body where there is a high distribution of sensory receptors. One is the hand or the hands, two is the lip and the mouth area, right. By the way, there are reasons why these two, evolutionary reasons why these two must be important, right, because I am at least evolutionarily, when I am trying to pick up an object, I need to examine what this object is, try to come to terms with whether it is you know, let us say it is thorny, it is a thorny fruit, what is inside is useful, I mean I like to eat it, but you know I cannot just put it in my mouth, and that is also sensed by the lip, like what goes inside is in some sense controlled by the, you know, lip, hopefully. So, which is why we like smooth things rather than, I mean, nobody likes to eat a, you know, sharp thing, right. So, that is the reason we like ice creams. So, anyway, so that is with haptic perception. So, what else? So, in summary, we have seen Golgi tendon organ, which is a nearly perfect force sensor or tension sensor. So, it is going to sense how much tension is there in the muscle, and joint articular receptors, these are believed to be sensors of joint angles at least in the extreme positions. It is not clear what their function is or how, if they are present in all muscles, it is there more information, more research needs to be performed to understand their function better. And then there are cutaneous receptors, these are Pacinian corpuscles, Meissner corpuscles, Merkel discs and Ruffini nerve endings, and other things such as nociceptors, thermoreceptors, free nerve endings and such. So, all this information goes to, as I said, goes to the either to the dorsal nucleus of Clark or to the gracile and cuneate nucleus, and from there it goes to the somatosensory cortex, where it is in some sense where sense is made out of what that our conscious perception is happening in the somatosensory cortex. It turns out that, how do you know, by the way, how do we know where the information is coming from? If the somatosensory cortex is receiving information from a whole bunch of body parts, is it not? It is receiving from all the fingers and hand, it is receiving from the rest of the body, it is receiving from the legs also. It turns out that there is, within the somatosensory cortex, there is a map of which area is responsible for receiving information from which body part. So, this type of somatic map or a body map, also called as this feature of a body map within the somatosensory cortex and elsewhere. So, a body map within the brain is also called as somatotopy. Okay. So, using that, there is this information that is sensed. So, with this we will stop this class with this, and we will see in future classes the following topics: reflexes, how these proprioceptors themselves cause reflexes, and what are the various types of reflexes. So, monosynaptic, polysynaptic, oligosynaptic, etc. And what are the mechanisms of these reflexes? Reflexes are movements, reflexes may be movements. So, a response of the body to specific stimuli, how these are controlled. So, with this we will stop this class. Thank you very much.